In this video, we're going to explore the evolution of the eukaryotic cell from prokaryotes. We're going to see the origin of eukaryotes. And this picture kind of tells it all. We have an early cell, and then there were some events that happened, such as folding in of the plasma membrane, and then there were some interactions between this weird folded cell and some bacteria that existed at the time to eventually lead to the more complex cells that we know today with mitochondria and chloroplasts. So to go from prokaryotes to eukaryotes, there were two main events that were involved. So the idea is that eukaryotes evolved from simpler prokaryotes. The first event involved is the infolding of the plasma membrane. So here we have an ancestral prokaryote, no membrane-bound organelles. It has a plasma membrane, that's the only real membrane it has, some DNA and cytoplasm, and that's about it. And over time, the plasma membrane started folding inward, so it was an inward folding of the plasma membrane. And that infolding eventually led to internal membranes. So we now have internal membranes in our cell. So we have membrane-bound organelles. We have a nuclear envelope over here, the ER, the endoplasmic reticulum over here, and then we have our DNA found within the nucleus. So now we have our membrane-bound organelles. But if you look closely, we don't have all our organelles present. You would have noticed that we're missing a very important one or two very important ones. So if this is an animal cell, we're missing the mitochondrion only. If it's a plant cell, we're missing the mitochondrion and the chloroplast. So let's go take a look at the other process involved. The first process was infolding a plasma membrane to form things like the Golgi, the ER, and the nuclear envelope. The next process is going to explain how the mitochondria and chloroplast came to be. So now we're going to take a look at how the mitochondria and chloroplast came to be. This is our second process involved. It's called the serial endosymbiosis concept. And so serial endosymbiosis works something like this. Here we have our ancestral host cell. This is a cell formed by that infolding of the plasma membrane. And over here we have an aerobic bacterial cell. This aerobic bacterial cell is a bacterium. And what it does is, we call it aerobic because it can use oxygen to extract energy from molecules in the environment through the process of cellular respiration, or at least a process similar to cellular respiration. Now what happens is, when a cell wants to eat, it goes near its food and essentially folds inwards to bring the food inside. And so that's essentially what happened. The ancestral host cell went near this aerobic bacterium and brought it in by pinching the membrane inwards. So this is part of the host cell membrane here. And so we brought that cell inside, and this was supposed to be our food. But what happened was we didn't break it down for some reason. It was actually an advantage to keep it within us for the ancestral host cell, because now it had an organism in it that was breaking down molecules to give it more energy. So it was getting more energy. And also this bacterial cell, well, it now had the advantage that it was protected in this ancestral host cell. So eventually over time, they just stayed together and worked together, and over time these bacterial cells became our mitochondrion, or our mitochondria. Our mitochondria are no longer bacteria, but they become part of one cell itself. Now, if a cell only took in this aerobic bacterium, it might have been an animal cell or a fungal cell somewhere along the line, or some kind of protist that does not have chloroplasts. But some of these cells, they went to consume more bacteria. And if they consumed a photosynthetic bacterium, the photosynthetic bacterium that was thought to have been consumed was a cyanobacteria. Over time, that cyanobacterium became the chloroplast in plant cells and certain types of protist with chloroplasts. So this was a photosynthetic cell that was able to convert solar energy into chemical energy, and in that process released oxygen into the atmosphere, which is probably why this bacterial cell was able to had the advantage when it was able to use oxygen to extract energy. But the idea is that the ancestral ancestral host cell engulfed some bacterial cells that became part of the ancestral host cell over time. And we call that endosymbiosis. Basically, you take in taking in a symbiont, taking in something that will work with you. So you might have remembered from grade 9 symbiosis, like mutualism, parasitism, and commensalism. Well, this might be an example of mutualism with these two organisms working together. But again, now our mitochondria are no longer bacteria, but they are part of our cells. So just to clarify some terminology, again, eukaryotes evolved from simpler prokaryotes. The bacterial cell that was engulfed is called the endosymbiont. The host is our ancestral host cell, the larger eukaryotic cell that engulfed it. So the advantages that were provided by engulfing the aerobic bacterium is that we could actually use oxygen now to extract energy from chemical molecules like sugar, and oxygen is now abundant in the atmosphere. So now that there's oxygen to use, it's probably an advantage to have an aerobic bacterium living inside of you. The advantage provided by swallowing the cyanobacterium, that photosynthetic bacterial cell, is that you could convert solar energy into chemical energy stored within certain molecules like sugar. So you could basically do photosynthesis. And over here you could basically do cellular respiration. So the idea is that some cells swallowed these aerobic bacteria that became mitochondria, and that's all they swallowed. And others might have, after they swallowed this mitochondrion, swallowed the cyanobacterium, and that eventually became chloroplast in other types of cells. The interesting to note is that this cyanobacterium, this photosynthetic bacterial cell, it was the one that was doing photosynthesis and made an abundance of oxygen in the atmosphere for this guy over here to use, which is why, again, being able to use oxygen became an advantage in this new environment. So there is some evidence to actually support endosymbiosis. It's not just made up or a fairy tale. These are the main pieces of evidence here. The first is about the membranes of chloroplast and mitochondria. They're actually very similar to those of living prokaryotes today. They have a double membrane. The inner membrane, so if you look at the inner membrane is said to be the, let's say, ancestral membrane of the prokaryote that was engulfed, and the outer membrane is said to be the membrane of the host that swallowed it. Because, let me just show you here, as you swallow a cell, let me just actually draw this with a different color. Let's go with blue. As you swallow this blue cell, the cell membrane pinches inwards. And so what's going to happen is you're going to have something that looks kind of like this. Let me go back to my red here. Right here. So we're pinching inwards. And let's go back to my blue to swallow this blue organism. And the red is just essentially going to swallow the blue. And when you do that, you essentially have this red on the outside and the blue on the inside. And that's our double membrane. So chloroplasts and mitochondria have this double membrane system, and their membranes look very similar to prokaryotes that exist today. The other interesting thing is that chloroplasts and mitochondria have ribosomes, their own ribosomes, that are actually very similar to prokaryotic ribosomes, which is quite interesting. And then the other thing as well is that chloroplast and mitochondria have their own circular DNA that's similar to prokaryotic DNA. If you study chloroplast DNA, you'll see it's actually very similar to cyanobacteria. And then chloroplast and mitochondria, they also reproduce in a similar way to prokaryotes. They reproduce using binary fission, and they can do that independently of the main cell. So these are the main pieces of evidence that highlight how similar the mitochondria and chloroplast are to prokaryotes, and to support the idea of endosymbiosis, where ancestral cells engulfed these aerobic bacteria and photosynthetic bacteria. So one of the questions that's often asked is what came first, the mitochondrion or the chloroplast? So mitochondrion is singular for mitochondria, but if you look at this picture carefully, you can come up with the answer yourself. You'll notice that this is our infolding process happening here to create those membrane-bound organelles like the Golgi, the ER, and the nucleus, the nuclear membrane. But then there was swallowing of an aerobic bacterial cell. And then after that, the cells that did not swallow anymore, they were just heterotrophic. They're the heterotrophic eukaryotes. They might have been some protists of some sort, maybe. And then those could have eventually given rise to animal cells and fungal cells. But some of them decided to swallow these photosynthetic prokaryotes. And so those ones that swallowed the photosynthetic prokaryotes became the ancestral photosynthetic eukaryotes. So they could have been ancestors to plants. They might have been some protists, and that might have been with chloroplast, that might have given rise to other protists with chloroplast, or perhaps some plants as well. And so scientists think that the mitochondrion arose first because all cells have mitochondria, but not all of them have chloroplast. I should say all eukaryotic cells have mitochondria, but not all of them have chloroplast. So mitochondria were swallowed first. Following that, some decided to swallow chloroplast and others did not. And that's why we think that mitochondria arose before the chloroplast. And that's exactly what this image here highlights. So you can see that we have our tree of life with our domains and kingdoms on the tips there. We have our bacteria and archaea domains, which are prokaryotic cells. And then we have our eukarya domain, which is eukaryotic cells. And you can see that because all the eukaryotic cells have mitochondria, we think that chloroplast arose later compared to the mitochondrion along this timeline of endosymbiosis. So this is our summary of how eukaryotic cells came to be, with the infolding process first, followed by endosymbiosis, specifically of the mitochondrion first, and then some taking the chloroplast in afterwards. Along with these new abilities to build these energy-rich molecules and use oxygen to extract energy from it, came the rise of multicellularity. So basically now the ability to be a multicellular organism. So essentially after eukaryotes came to be, some of them started to live in colonies, and the ones that lived in colonies, they started to divvy up tasks and become specialized in different tasks, and eventually they became so specialized, different groups became so specialized, they forgot how to do everything else. So they had no choice but to live together. And that's one idea behind how the multicellular organisms came to be. So it was thought that maybe a multicellular protist somewhere eventually became some kind of plant, or maybe some kind of animal, or some kind of fungus, depending on the mutations and the transformations or the changes that happened in those organisms. So as you can see, protists are thought to be kind of those older organisms, at least the older protists. I mean, the ones that exist today are still equally evolved to the plants and animals and fungi, but protists are thought to have arisen first in that whole process of evolution. So the idea was that we had prokaryotes first, then protists, then the protist eventually gave rise to plants, animals, and fungi. So you can take a look at the tree of life over here. So we have our tree of life, and in our tree of life you can see we had some kind of eukaryotic cell that swallowed a cyanobacterium, for example, and that eventually became a chloroplast, and then eventually perhaps this organism here might have had enough transformations to become red algae, and then eventually form other types of plant-like protists, whereas this green algae here, it went through many other transformations and may have formed other type of plant-like protists, and may have even gone through other transformations to lead to the formation of plants altogether. So we have prokaryotes going to protists, then forming plants, animals, and fungi. But now you know how prokaryotes led to the evolution of the eukaryotic cell, starting off with the protist first. So you can read up on this idea in the Nelson textbook, page 61 to 62. You can attempt the practice problems on this slide. and do any of the questions that are on the learning goals that you'll be provided with via D2L. And then you can watch a few videos that goes over this idea in a bit more detail with some clever animations that will hopefully help you understand the concept a bit more.